Okay, we're going to begin. Thank you everyone for your cooperation. We're going to start with a brief introduction from Ella.
Thank you so much, Will. It's a pleasure to welcome each of you coming to the China Night. Many of you already attended the daytime meeting conference, so thank you so much for staying with us for this evening. Several things to announce. First, we have 11 roadshow companies. Later, going to have the pitch, and after that, they're going to have their stand on that side of the boat. So if you would like to talk with them, just go there. And our dinner starts probably around 8:30, right, Minghua? 8:30, and followed by line dancing. And if you are the VIP receiving our email yesterday, feel free to go to the third floor of the VIP room. Okay, I think we can just get started for roadshow. Yeah.
All right, perfect. Let's move on. So for our first company is Arromax Pharmaceutical. And our speaker is Professor Zhengbo. I'm going to leave it up to him. You have the stage.
Okay.
Quiet in the audience, please. Thank you.
I share the PPT. Okay, good evening, good night for everyone. I'm Zhengbo Song from Zhejiang Cancer Hospital in China. On behalf of all of our PI, it's our pleasure to present our phase 1b and 2 clinical trial to evaluate the safety, effectiveness, tolerability, and pharma profile of AMX3009 in patients with locally advanced or advanced non-small cell lung cancer having uncommon EGFR mutations. This is the background. All we know that PCC mutations account for about 12% of EGFR mutations, making them the second most common type following classical EGFR mutations. For these patients, there are no approved first-line or second-line targeted therapies in China. So there's a great need for this patient to have a EGFR TKI or targeted therapies. This is the AMX3009 background. AMX3009 is an oral EGFR TKI targeting uncommon EGFR mutations and is a small molecule targeted drugs designed to address PCC mutations. This is our study design. The study including phase 1b and phase 2. For phase 1b, 6 patients were enrolled and 3 with 14 milligram and 3 with 13 milligram. For the phase 1b, including stage 1 and stage 2. For the stage 1, we compare the 2 doses, one in 14 milligram and one in 30 milligram. For at last, the 13 milligram were confirmed as RP2D dose. For in the phase 2 stage 2, all of the patients were enrolled in the 13 milligrams. This is the result, swimming plot and waterfall plot. The result showed that in the 14 milligram, 12 patients were included, and we can see that 7 patients achieved PR with an OR rate about 75%. For the RP2D dose, 13 milligram, we can see that currently 19 patients were enrolled, and at last, all of the patients were with disease control, with a disease control rate 100%, and 17 patients were achieved PR with OR rate nearly 90%. This is the safety analysis. Totally, we can see from the table, the 13 milligram group didn't experience any drug-related SAE. Reported only 1 patient with a TRAE of grade 3. and had no TIE leading to treatment discontinue, and the safety is better than the 40 milligrams. We compare with another EGFR TKIs in China. For one was formatini with a phase 1b trial of the PACC mutation, another was alpha-tini, we all know. From the table, we can see that for the ORR rate, the AMX result showed a high ORR rate, and we can see at TIE grade 3 or more than 3, we can see at of the 3 the EGFR TKIs, the AMX showed a better, a lower toxicity compared with another EGFR TKIs. This is the same as OR. Totally, 9 patients were enrolled in our clinical trial, and we can see that from among the 9 patients, 7 patients showed achieved a PR with an OR rate of 77.8%. So the AMX result showed a better effective for the CNS metastasis. This is in summary. We can see from the result of the phase 1b and phase 2 study, the AMX showed encouraging result of encouraging OR and DCR and the efficacy and lower toxicity. The phase 2 stage 2 study is ongoing, and randomized phase 3 study compared with the chemotherapy will be studied in this year. That's all. Thank you very much.
Thank you, thank you. Up next, we have Ray Lee with Teclison. Ray Lee. 应该是这个，应该可以。您的 PPT。
在哪里？ Hi, good evening. This is Ray Lee. I'm the CEO of Teclison. Today, I'm going to talk about our new immunotherapy approach and to treat solid tumor. And we have the main focus by expanding the anti-tumor T cell. Okay. One of the major hurdles for immuno-oncology is because the majority of the patients do not have a sufficient anti-tumor T cell. Even if they have some, and even if they have some anti-tumor T cell, they tend to be exhausted. And this is because of the chronic exposure to the tumor. And what we have done and trying to overcome this is a problem is using 2 strategies to induce tumor necrosis. And this is will result in release larger amount of the tumor antigen and then generate a lot of anti-tumor T cell. This is a kind of a bypassing the problem of a low anti-tumor T cell and also the exhausted T. And then so those expanded T will be able to synergize and activated by the immune checkpoint inhibitor and really increase the efficacy of anti-PD-1. So we use 2 strategies. One is called TATE, which stands for the transarterial theraplasmic embolization. And the other one is called THIANA. And both approaches would able to induce a very rapid and strong tumor necrosis. And this is a lead to expansion of the anti-tumor T cell by at least 100-fold. And then the expansion is correlated with clinical response. And then, as I said, those T cells would be activated and synergized with anti-PD-1. So we have a fairly robust efficacy. We have established pretty much a proof of a concept clinically in the hepatocellular carcinoma who are refractory to immunocheckpoint inhibitor. And then we were able to achieve 55.6% of the response rate. And in this is a difficult to treat population. And then this is even higher than the first line, if you recall. And the first line immunotherapy, such as the Nivo plus Ipi, could only achieve a 30-something response rate. And this is our clinical data in the phase 1. And we use this intermediate stage of liver cancer to test our TATE strategy. And then the main point is to establish the fact TATE is able to induce tumor necrosis. As shown in this is a waterfall plot on the right, and you can see we achieve a 60% CR rate. So I want to emphasize this is a CR. So this is how robust the tumor necrosis effect is. And then in the phase 2 clinical study, which is we just presented a couple of days ago in the GI ASCO, and with the first 18 patients, we have a 10 response in the immunotherapy refractory population. And this is a situation can also apply in other solid tumor with the liver metastasis in other clinical trial. And this is one of the represented patient, and definitely PD-1 refractory. And then you can see the liver lesion has significant undergo necrosis after the treatment. But the most exciting is the person has an extrahepatic adrenal metastasis. Before the treatment has a 6 centimeter, and after treatment, dramatic shrink in the size. And the person actually, I believe, achieved a CR, even there's still some residual lesion. And then because you can see the right panel, the alpha fetoprotein completely normalized for more than a year. So I believe this patient is cured even after it has been treated with multiple line and also refractory to immune checkpoint inhibitor. So, and how can we explain this is a phenomenon? And we believe it's because of the expansion of the anti-tumor T cell. So to prove this, we isolate the PBMC to conduct the next sequencing. And we see not only the expansion of the existing T cell, but also generation new anti-tumor T cell. So this is explained why we can have such a robust abscopal effect and then make the patient do very well. And then this is the current clinical trial we're conducting, not just on advanced HCC, but also did in colorectal, non-small cell lung cancer, gastric, and then basically is as long as they have a liver metastasis, we were able to apply our technology combined with anti-PD-1. And then the market for solid tumor is huge, is easily supersede 10 billion. And then we can also take advantage of the fact, just isolate the anti-tumor T cell from PBMC, do an ex vivo expansion. Remember, I just mentioned in vivo, by TATE alone, we expand 100-fold. By taking the PBMC out and then do the ex vivo culture, we expand another 100-fold. So this is a sequential approach that will be able to achieve 10,000-fold of the anti-tumor T cell expansion, and you can give back to the patient. So this is a completely new type of autologous cell therapy without the need of the genetic modification. We don't need to put the CAR T vector in those T cells. Just isolate those as a PBMC, direct expansion, and put back the patient. Simple and lower costs compared with the current approach. And then we are currently in the process of raising Series B with 30 million. and then trying to move into phase 3 clinical trial. This is our management team and all with major pharma experience. And then finals, this is the final slide. I want to convince you and then also convinces a potential investor why it's worthwhile to invest on Teclison. And then we create value for those companies who make anti-PD-1 antibody. And in 2 years, you see both the Merck and Keytruda or the BMS Optimal is going to be run out of the patent. So there's a 40 billion gap and remain to be compete. So there's a lot of a competitive player out there who is going to win. And I don't believe each antibody is going to be better than the other. So how any of the companies going to win the race is depends on their combination partner. And I hope I've convinced you the Teclison's strategy is going to be the best to help them win the race. I'll stop here. Thank you.
Thank you, Dr. Lee. Next, we have Han Lee, CBO of Pulse Medical Technology.
Thank you. All right, hello everyone. Happy to hear and good evening. My name is Han. I am the CBO at Post Medical, and we are a commercial stage medical device company specialized in structural heart disease with interventional therapies. So very high level company overview. We founded back in 2014. We have our own R&D center, manufacturing facility, integrating both medical and industrial engineering. And we also have international R&D team across the US, Europe, and China. And we are currently in the pre-IPO phase, looking to list on Nasdaq by the end of this year. So very high level company development milestone. Again, we started about 11 years ago, starting the R&D journey. We have expanded our product pipeline, as well as commercializing our very first product, left ear LLA oculator, with Johnson & Johnson partnership in China. So that's where we are at as of today. So here's a photo back. We signed a contract with Johnson & Johnson where they are responsible for sales and marketing, and we are responsible for manufacturing and delivery in China. And we are looking to continue the partnership beyond China, getting into Europe and the US. So here is a summary of our pipeline. Again, we have the left ear being marketed already in China, potentially also getting approval in Europe at the end of this year. We also have a pipeline of the PFO occluder, the coronary sinus reducer, as well as a LV partitioning device. We have a very experienced leadership team with our chairman, Ron, with over 20 plus years in sales and clinical practice in the cardiovascular medical device space. We also have a very deep industry expertise teams, including our VP R&D, manufacturing, marketing, as well as our research institute. We also have a very strong KOLs, as well as our top chief PI, professors, advisors across top hospitals. So here's a table of a summary of our global patent, where we file 142 patent already. 86 of them it's already approved, and we are continuing to span out our international PTC map. All right, here's a high-level summary of our product pipeline. So we got our first left ear marketed already with Johnson & Johnson about 1.5 years ago. We have our future generation of the left ear coming to the market. Along with that, we also have a PFO pipeline, stent product, and then also delivery system product. So we are going to have additional product coming into the market, increasing our sales, not just in China, but then also in Europe and the US. So again, our left ear product, it's first in class in terms of it's being a double disc plug. It's It's easy to retract repeatedly, so reduce the complexity during the operation. Our PFO oscillator, just going through this high level, again, very similar design. It works for various type of PFO structures. Again, it's easy to retract and then reduce the complexity during the surgeries. All right, so I just want to call out another pipeline we have here. One of our competitor recently raised a $23 million Series B. So we actually have a similar pipeline going through preclinical and potentially achieving very strong data in a similar space. So this is another pipeline we have in the portfolio with recent traction and momentum in the space. All right, so here is two more slides. So here is our financial focus just based on our contract with Johnson & Johnson. So we were able to break even, become profitable in 2025. And then in the next 3 years, we are going to double our revenue and profit every year. And this is more of a conservative forecast just based on our contract with Johnson & Johnson in China. But if we get approval in Europe later this year, it will add on this. And then with any additional pipeline being approved in 27 and 28, it will also add to the financial forecast here. So very last slide here, we are in the process of closing our pre-IPO funding. And again, we already started the IPO process with our investment bank, auditors, and law firms. And we are looking to get listed by the end of this year. So that's all the presentation. Thank you.
Thank you, Dr. Han. Up next is Dr. Kartik Krishnan. Welcome.
Actually, I am not Dr. Kartik Krishnan. He is our CEO. He is here in the back. I'm Brent Sorrells, the Chief Operating Officer for OncoNano. Thank you for having us. Our lawyer really loves this slide, but just so you know, none of our assets we're talking about have been approved by any regulatory authorities. We at OncoNano are advancing a new class of targeted cancer therapies with our onboard platform. This platform is made up of pH-sensitive polymers that form micelles to encapsulate payloads. These payloads exploit the acidic microenvironment that surrounds most tumors. And this encapsulation protects those payloads, especially when they're potent or toxic, from any kind of systemic exposure. We announced last week a deal with Gilead. This collaboration is valued up to $300 million for the life of the collaboration, but it is for us to encapsulate one, possibly two of their assets that they currently have in development. And additionally, this platform has already been tested in humans through a phase 2 study using a fluorescent molecule for imaging tumors. A little more about how it works at normal biologic pH. Our polymers form micelles. Any of the variety of payloads we can carry that are tied to those polymers are safely encapsulated on the inside of that micelle. It is an almost instantaneous release of the payload once it hits that tumor microenvironment. Our polymers are fine-tuned to only release once they hit that very unique acidity around the tumor. And it is a very precise response. The range of our transition pH for our polymers is 0.25 of a pH unit. So this is a quick look at our pipeline. Our lead candidate is ONM-421. It is carrying a payload of MMAE. It is currently in preclinical work doing IND-enabling studies. ONM-423 is our second candidate, and ONM-067 is kicking off right now with our partners at Gilead. Pegcetacyanine is the fluorescent payload using our onboard platform. So this is a phase 3 ready asset. It has been our proof of concept for the platform, showing very precise release at the tumor. And then one other product we have in our pipeline, it is unrelated to onboard, but it is also a polymer drug conjugate. It is different in that both the payload and the polymer activates STING, and our polymer works to prolong that activation. That asset is currently in phase 1 studies, and it is being administered in combination with Regeneron's Libtayo. So we have some preclinical data on our 421 drug candidate that we'd love to share. We also have clinical data for our 501 that we would also be happy to share, but time permitting, I'll just give a quick summary. So we think we have a great way to deliver toxic payloads directly to tumors without having systemic effects. We've got evidence of improving therapeutic index on drugs. And I think the de-risking we've done through our pegcetacimab program really leverages all of the experience we have in-house. And this is kind of proven out with our ability to collaborate with companies like Gilead. We are looking for a $40 million Series C and would love to chat with anybody interested in learning more. Thank you.
Thank you. Next, Zhu Lun from Excalibur Point Therapeutics.
You get my presentation? Can somebody get my presentation up there? Do I have to do it myself? That's not me. All right, cool. Thank you. All right, so our company is called Excalibur Point. The name is actually an amalgam of 2 words, Excalibur and Point. Excalibur is a big, powerful sword in Northern European legend. Point, as you know, it means targeted delivery. So our company is a T-cell based therapeutic company, which means we utilize the power of T cells. But it's not a blunt tool, it's a targeted tool. So we want to make the sword to point to the right target, which means tumor and autoimmune diseases. Okay, so that's the story behind the name. Okay, so here are the 2 people behind the company. This guy, my partner Lei, he's stuck in China because he's not getting his visa approved in time. So I'm standing by to do this delivery. Lei is, both of us actually work at I-Mab. All right, so I-Mab was some of you may know this company. It's one of the first generation monoclonal antibody company in China. I was the CFO for I-Mab about 6-7 years ago. Took them public on NASDAQ. We were one of the first few Chinese biotech companies to go public on NASDAQ. Lei was one of the co-founders of I-Mab. He was one of the students of Jingwu, who was the founder of I-Mab. So after I-Mab, he went on to join Lopu Biopharma as the head of their VP for R&D. And at there, he did a whole bunch of things. One of which was to execute R&D to establish a TCE platform with a couple of good assets. So last year, he and I worked out a deal with Lopu Biopharma. We did one of the first Chinese local newco with our company. So the deal was we took the TCE platform with the 2 assets out from Lopu. We gave Lopu $10 million and 10% equity in our company. That's the consideration. And then simultaneously, we raised $41 million. There's a slide after this I'll show you. $41 million from the market to pay for the $10 million for the upfront payment and also for the operating needs for the new company. So why this is a first Chinese newco? Because the assets are from China. The money is also from China. And then the team, also Chinese. So this is slightly different from what you guys usually saw in the craze of newcos in 2024 and 25 because this was not a US VC coming to China, swooping in good assets and putting the US-based management team. Okay, so slightly different. So we are in China, but doing global deals in the future. The key is utilizing the Chinese efficiency and cost efficiency and the speed, as well as the maturing ecosystem to get the assets into transactable stage as quickly as possible. I'll skip my ads. Some of you may know me, but I can talk to you about my creds later. So this is the money we took in our seed round. Hongshan used to be called Sequoia, but now they're called Hongshan, Yuan Bio, and Apricot. They are all very active Chinese, tier one Chinese healthcare VC. So they funded this NewCo, and then these guys on the bottom, 5Y, Cohen, Medfine, Honey, they collectively put in $11 million. So we raised $41 million, which I believe was one of the largest seed round in China for the NewCo. I'll skip this slide. This is our team. I think we represented an interesting way of putting together NewCo because all of these guys work with us either at IMAP or Lopu. So we know these guys can hit the ground running from day one, and they are battle tested. Okay, so I don't have time to go through all of the scientific slides. So suffice to say, our theme is TCEs for solid tumors and autoimmune diseases, but solid tumors first. TCE for solid tumor is actually a story that's just beginning. There are only a few deals happening in the market, and there's only one drug approved, TCE approved for solid tumor, and that is that Tolerability. It's an Amgen drug, conditionally approved in 2024 for small cell lung cancer. There's a lot of data here, but I'll tell you one big number is they have a 40% ORR, which is for heavily pretreated patients, and which was a great number. But there are a few problems with it, and we're trying to tackle those problems by working on a second-generation TCE. So here's what we're working on. So we have 3 proprietary technology platforms. This one is what we took out from Lopu. It's a TOPA, means co-stimulation. So we have 3 targets. It's a tri-specific setting. So there's the CD3 for the main signal, 4-1BB for the co-stimulatory signal, and then there's the TAA. We switch to TAA, we get a new asset. So our first asset is a validated target, which is DLL3. So the difference between us and the Amgen drug is we have the 4-1BB arm. Now, 4-1BB is an interesting choice for co-stim because there are 2 other choices. One is CD20 and the other one is TD2. We believe 4-1BB has the advantage in these 2 areas. A, have a higher efficiency of turning cold to hot tumors. And B, this one is more important, eliciting long-term memory by recruiting naive T cells from peripheral cells into tumor. I won't go through the science, but this is something that we believe in, and we have data to back it up. Our second platform is a T cell immune shield platform. So the thesis is that a lot of the times TCEs don't work in the tumor because the microenvironment, tumor microenvironment is all messed up. So we have a multi-specific molecule in PCC stage by activating immune system as well as modulating tumor microenvironment to make it more optimized. Lastly, we have a TCE prodrug platform, which is similar to what Genix has, but we have a different peptide and the AI to drive the peptide linker optimization. This product is currently also in PCC stage. We are using a previously undruggable target to make it more druggable or druggable. All right, I'll skip the next few slides and jump right into our pipeline slide. Okay, here's our pipeline. It's actually a partial shot of our pipeline. This is our first drug, which I talked about, DLL3, 4-1BB, and the CD3. we received the R&D clearance in China last year, and then the product right now is rapidly advancing through phase 1 trial in China. Dr. Lu Shun is our PI, leading PI in China, in Shanghai Chest Hospital. We are going to be release our first set of clinical data with safety, PD, marker, clinical signals, initial clinical signals, and RP2D by the end of this year, around Q3, Q4. This is our second asset from the same platform, the co-stim platform. This is a different TAA, CDH17. As some of you may know, it's a very clean target for GI tract, very highly expressed in colon and gastric. Third asset is a STEAP1/STEAP2. We're probably going to use STEAP2 for prostate cancer. Same setup, CD3, 4-1BB, and STEAP2. I have one more slide and I'll end. Okay, these are the clinical trial plan for our product. I'll end on the last slide so that you remember what we do. Okay, so we are a new generation Chinese newco, which means A, we're not a single shot company. We have multiple shots on goal. We have a platform. Actually, we have 3 platforms. We throw off multiple assets in the hope that some of them may actually have good phase 1 data. Secondly, we are a built for BD kind of company. So after we generate phase 1 data, maybe early stage phase 2 data, we'll be actively looking for ways and opportunities to strike partnership deals. Third, most of our clinical trials are done in China. So the point is to rapidly advancing these things and generating data. Probably our assets are probably 6 to 9 months apart. So by 2 years, in 2 years' time, we'll have two to three clinical stage assets. Hopefully, we'll be able to do 1 BD deal by then, and we're looking at IPO in about 2 years. I think that's it for us. Thank you very much.
Okay, thank you. All right. Next is Harry Lee, Wilmington Pharmatech.
Good evening. My name is Harry Lee, and I'm the president and the CEO of Wilmington Pharmatech. And this is Wilmington Pharmatech is based in Wilmington, Delaware. We are a science-driven CRDMO. Too fast. Got 6 minutes, right? We started back 2003, so I'm 23 years old now. And when we started, it's what happened to this one? Okay, click. Sorry about it. And when we started as a solid-state chemistry lab, means we're making crystals for the new drug. And ever since, every year we keep expanding. And recently, we added the GMP pilot plant, commercial plant in Delaware, and the high-potent production suite. And then we acquired the Explorer separation purification for peptide and ADC last year. And with new capital investment from QWell, we continue expanding in Delaware. Now this has 3 plants actually in Delaware, 1 in China, Suzhou. So our facility, we have this beautiful campus acquired from old DuPont with 54 acres expansion there. What we do mainly is basically 6 blocks and 3 main areas. And the chemical synthesis, anything you need, we can make it. And crystals. If you have any issue about your drug, we can improve that. And anything related to the analytical related and the large-scale purification. So our sweet spot actually get the new drug into IND very fast. We claim to be the fastest, and I don't know if anyone can do faster. And then we help the drug get all the way into NDA. All these years, we actually did more than 200 new drugs and pushes through, and 8 of them get NDA approval. Now, the first thing usually we do is we help the biotech company in US mostly. They develop the compound and they had some issue for the early stage of their drug candidate. We actually can improve the physical property and get the new drug candidate. Now, in case of the complex synthesis, we handle very complicated molecules, highly potent. We just did another 42 steps, 67 kg synthesis. And making the crystal actually is a work of art, and you never know, and that's no actually typical book you can learn. We have very special skill, and the main thing to get the better crystal is to get a better solubility of your drug, better bioavailability, and stability. So we made more than 160 patents. All these new crystals actually become better drug candidates as well. Now, with the drug candidates in hand now, we can make your drug at kilo scale and to the pilot plant, commercial scale, and basically seamlessly develop from phase 1 all the way to commercial. This is the kind of the real old DuPont type chemical plants, and we actually build it brand new and with the high potent, and we basically can handle any potency of the compound. And the purification, because a lot of compounds nowadays, ADC peptide, you cannot get crystals. That's why we get into this large scale purification. This is actually old Novacep technology. And we have, if not thousands, hundreds methods and columns. We have all the on-site analytical instrumentation. Now, what we also have another site in Suzhou, China, utilize what's good at China and the raw material. We actually emphasize in working on this N-1. We do most of the work in China, saving the cost. And while we do the final production, final QC, QA, and large scale storage, in case something happen, and we actually store years of the supply for you. This is the just green piece. You can see we bought all this land there. So that's the reason we still continue expanding. And what we see is a wave of the new compounds from China. I think we can help increase the value and make it more attractive to the big MNC. So we help you not just the IND, but increase the value pretty dramatically. So what we do is chemical synthesis, crystals, purification. Thank you.
Now, Sean Zhuang from Cardio Story.
Can I have the stick? Thank you. Good evening, I'm Sean, CEO of Cardio Story. Cardio Story is the Delaware company with a manufacturing facility in China. It's my pleasure to introduce these breakthrough solutions for heart failure diagnosis and management. Heart failure is a big burden. So we know that as the people getting older, various kinds of heart disease develop into heart failure. If we look at the data, it is now the #1 cause of hospitalizations in aged patients. And today, I believe that many of the audience here may live over 90 years old or even 100. And in that case, over 30% of the population will have heart failure. So this is a big burden. And once they have heart failure, they are repeatedly sent to hospital again and again. So how can we improve this condition? Let's make an analogy. Today we know that we can control diabetes quite well. We can just measure the vital signs and keeping the blood sugar level in optimal optimal range, then it well controlled. Similarly, if we take a look at heart failure, if we can measure the intracardiac feeding pressure, that means the pressure inside the heart. If we can measure and control this pressure, the condition of the heart can be controlled. However, unfortunately, there is only less than 1% of patients that can receive close monitoring of this pressure because the device now are invasive or implanted. So we need a new tool, a new device. It should be non-invasive, it should be easy to use, and then it can be used at millions of homes and hospitals. So here is our solution. Cardio storage solutions are using patent sensors and algorithms to calculate the feeding pressure. The sensors, typically they are listened to the heart from the patient's back and from the front, not just the heart sound, but also the inaudible part, the sub frequency. Together, we translate this vibration into the feeding pressure in the heart. And this technology has been granted breakthrough designation by FDA, and we were the first one to do that. And the test only takes 2 minutes. They don't need a wearable. They only need like a test per day or twice per week. How good is the device? Is it ready for commercialized? Yes. We have over 5,000 patients tested by this device, so the usability is okay. And we also look at the performance. We had quite a lot of data showing that in emergency room, this device can support diagnosis of heart failure in a good accuracy, 91.5%. This is excellent. If we take a look at another situation that this device can evaluate heart failure feeding pressure in surgical room for heart failure patient, the accuracy is 88%. This is also very good. And the good performance brings us to good business opportunity. We can see that many of the hospitals, they need this device. So our first market is sell to the device to the hospitals, and it's going to generate continuous revenue from disposable part. And the second business model, we are going to sell the device to home use, and then it generates a subscription fee from the service. In the future, this device will combine with traditional EKG, and it's going to be an up version, a premier version of EKG, and in the future, it's going to replace most of the traditional EKG. Here are some of the spotlight. And in the last several months in 2025, we were awarded the best startup that using AI innovation in ICU. And we also supported by some of the centers and accelerators. The team, and we have experienced meta innovators, and I have more than 10 years served as the global marketing head or the public listed company selling cardiovascular device to over 80 countries. And I also have many years of R&D experience. My co-founder, he is a PhD and scientist in fiber optic sensor. He is the pioneer that using fiber optic sensor to detect the human vital sign and make it commercialized. We have also very experienced advisors covering clinical, regulatory, and reimbursement. So we are now doing our pivotal study. It's almost complete. It will be complete in the coming 2 or 3 months. We need 1 million so that we can get CE marking. We need another 4 million so we can get FDA approval and ready to generate sales. Welcome to join us. Your investing of this will help us to bring this breakthrough technology to millions of homes and millions of hospitals. Thank you.
Wang Tiansheng from Henan Micro Biomedicine.
Thank you.
Good evening, everyone. My name is Tianxin Wang from Immunotherapy Biomedicine. Today, here we go. Thank you. Today, I'm going to present a new molecule called TM471, which has innovative and high selective BTK inhibitor. BTK inhibitor blocks BTK signaling pathway of B cell. And this pathway was well clinically validated by a few FDA approval as drugs. The limitation of approved BTK inhibitor, including in the lymphoma area, poor selectivity, and always some off-target associated toxicity. In the autoimmune area, usually the BTK inhibitor will have cardio side effect as well as liver tox issue, like we see recently with Sanofi drug. 471, we are targeting some unmet medical needs in the BTK inhibitor area. In the lymphoma area, we are doing marginal zone lymphoma. In the autoimmune area, we are cover 4 areas. First is autoimmune encephalitis. Our compound is the first compound being developed in this area. And also we are doing multiple sclerosis and chronic spontaneous urticaria as well as membranous nephropathy. The story of discovery of 471 was published last year. I'll not go to detail. This is a very well-balanced molecule in terms of activity, selectivity, PK, as well as safety, which made it suitable for long-term chronic use. The advantage of this molecule is higher exposure and also a large therapeutic window, and which was already proved by preclinically with a large MTD and also a very wide safety window. And also, most importantly, it's already proved by clinically. We went through phase 1 clinical already, and with no adversary effect was observed. I'll go to some detailed data of 471 in terms of BTK activity. We are listed here as a close comparison of some approved inhibitor, including zanubrutinib, compared with TM471. They have same potency. However, in terms of target EGFR, BLK, the TM471 is give much good selectivity, much better, and also for the safety, heart-related safety enzyme for HER2, HER4 of TM471 has a much larger window compared with zanubrutinib, and also in HERC, which is like a very high IC50 value. All these data indicate a much safe heart safety. And the TM471 also demonstrated more than 4 times MTD compared to zanubrutinib, which is currently best-selling drug. And we also demonstrated 3 times brain permeability than zanubrutinib. TM471 achieved faster and more sustained BTK occupancy for BPMC and spleen. So also as well as in terms of exposure, showed 1 order of magnitude higher systemic exposure, making it a stronger and more durable clinical efficacy. In terms of in vivo efficacy, you can focus on the right side. TM471 compared with zanubrutinib, 7.5 milligram, and 471 is we use 3.7 milligram already achieved similar efficacy. And if we use 7.5 milligram, we cleared all the tumor. TM471 showed a comparable efficacy compared with renobritinib by reducing microglial activation and CD4 positive cell infiltration, and making it suitable for treating autoimmune disease with CNS indication. And also, TM471 had superior therapeutic efficacy compared with renobritinib by skin lesion score reducing, and also the number of views, making it a potential more effective treatment for chronic spontaneous urticaria. In terms of safety, we already went through phase 1 clinical trial. No dose limiting toxicity occurred in all cohorts, and also no significant change in cardio, liver, and kidney function. Currently, we are focusing for the 4 autoimmune disease area, and a couple of lymphoma area. And also besides this, we still have a few candidate program, and some of which already undergoing IND-enabling study. In conclusion, I hope I can show here TM471 is positioned as a best-in-class BTK inhibitor, and which is supported by strong science and early clinical validation, and we welcome partnership opportunity. Thank you very much.
Thank you. Wang Yu from Idea Pharma.
Okay, good evening, everyone. So my name is Yu Wang. I'm from ID Pharma from China. So I will proud to present you our exciting program, ACC017. It's a novel third generation instinct for HIV AIDS. So yeah. So our company, ID Pharma, the name came from, I have a good idea. So we are motto is ideal care for HIV. And our company is founded in 2009, IPO in 2020 in Shanghai stock market. We already have 2 products get approved, Enovarin single tablet and Enovarin combo. So we position ourselves as a best leading Chinese pharmaceutical company in HIV disease area. So next slide, I will present you this very exciting program, ACC017. It's a novel third generation instinct for HIV. So right now, yeah. So we are offering the licensing and new co-partnership opportunities. So the territory is North America and Europe. And we are currently offering licensing and partnership, new co-partnership as well. Molecule is third generation instinct indication, HIV and AIDS. Timing, why we team up together right now? So the timing is very important. Currently we already running global phase 3 clinical trial, kick off in Q3 last year, and we will run the FDA IND enabling in this year Q3. We will run multiple national clinical trial, United States, China, as well as South Africa. So I'm also to present you some unique selling points and competitive advantages. So the single tablet, ACC07, novel instinct, is a potential optimized alternatives to dolutegravir and beta-gravir. It's also best-in-class candidate. We also have the combo. Combo is a potential alternative to Biktarvir and Dovato. The benchmark market size is about $25 billion US. And the strategy differentiation, the advantages are very clear. A rapid virus suppression, 65% faster compared with the current approved instinct. And dosing efficiency, lower effective dosage compared with competitors. Resistant profile is also better. Reduce the drug-drug interaction, fever drug-drug interaction risks. And IP protection coverage onto the 2040s. So this slide also shows you a history of HIV integrase inhibitor from the first generation integrase developed by MSD, a second one from GSK and Gilead, Givoya and Biktarvy. So the 2024, the Gilead's Biktarvy alone, the sales is reaching $13 billion US. So the market size is quite big enough. We calculate is about combined in total instinct for HIV is $25 billion US per year. So that is big enough to support our program in United States. And to compare with the current approved one, our compound binds to the integrase target more tightly. And also this slide show you a pivotal phase 3 study design. Actually, this phase 3 study is already kicked off in Q3 last year. So in total, we recruit the 60 patients. So far, recruitment is quite smoothly. We already recruit 150 subjects in the study. Yeah, the leading clinical sites, including Beijing Ditan University Hospital, Guangzhou 8 Hospital. So, and why we need a new instinct? The rationale including the better efficacy. So this slide show you efficacy data. You can see the right line. So in our compound, 40 milligram is efficacy is comparable to the Biktarvy 100 milligram efficacy. So, and faster virus suppression. In our compound, so the phase 2 clinical trial shows very solid and robust data, very fast virus suppression. 90% virus have the virus suppression in 4 weeks. And also the rationale why we need new instinct, including the limited cross resistance as well. The mutation with another approved instinct doesn't work in our compound. So the mutation in this remains susceptible to our compound. And another risk of the drug-drug interaction, our compound is stay unchanged form in the human plasma with minimal metabolism in the liver and lower drug-drug interaction risks. And also the rationale including the less side effect, cardiovascular effect, as well as obesity and cholesterol-related side effects. So our compound also shows the better safety profile, smaller dosage. So the key home take message, the advantages of ACC017 is obvious. Virus suppression is faster, within 4 weeks. Resistance profile is better than the current approved one. Drug-drug interaction, lower drug-drug interaction risk. Safety profile is better, and dosage is lower. So the timeline, 2025 Q3 phase 3 clinical trial kickoff in China. And this year Q1 we will conduct FDA pre-IND communication. 2026 Q3 FDA IND enabling. It's a clinical trial ongoing. So this is my contact. So welcome you to reach out to have more details. What's more, we have more long-acting agent for HIV treatment and PrEP prevention upon your request. Thank you very much for your attention. Yeah, thank you.
Thank you. Hang Qiling from Koba Life Pharmaceutical.
Okay, thank you, organizer, for the opportunity to present our pipeline. So we have promising blockbuster PD-L1 VEGF bispecific antibody, which is in a clinical trial. So first, I will very quickly brief our company. So our company is established in 2018, and we have one genetic drug approved 2024 and generated revenue to support our company for salaries. But we do need the money to develop the clinical trial, particularly for this big marketing like in bispecific antibodies. Our strategy is based on the success in clinical and also success in sales. How can we success in clinical? We build our own clinical team to design and develop in clinical so that we can trust our data. We also do the partnership with like a large sales teams company to ensure we have light investment, but we have good sales. Okay. And our team is a seasoned management team. So our CEO is very young and very energetic. Our pipeline, so we have one genetic drug, which is approved in China and also just got approved from Indonesia. We have the bispecific antibody, which is I'm going to talk about. We also have 3 other small molecules in clinical development. So as we know, last year is very busy and very good news for the bispecific antibody. Started with AK112, get approved in China. However, we still need to show OS get achieved. And so the race is not ending yet. And our drug from the clinical from the molecular design and also preclinical data, and we believe we still have a chance to be the best in class. So this is our drug molecular design, and we use the BAFA and fused with PD-L1 VHH, and we retained the FC end of the BAFA so that we can return the ADCC effect, which is recruiting the anchor cell to help to kill the cancer. And also because of this fusing together, so increase the VHH PD-L1 by 13 times. So which means we are expecting the better activities. And the third one, because this structure is a 2+2 symmetric structure, is very easy in terms of like a CMC. As we know with the antibodies, the monoclonal antibody probably is difficult, but the bispecific antibody is much more difficult in terms of CMC. So our product, we scaled, we switched the manufacturing facilities, and we also get like 3 years of stability studies. So our process is very robust and very easy. And we have clinical study for the monotherapy, I finished the phase 1A and 1B, and we showed very excellent safety. And we also in the combo with ADCs or chemo, which already showed very good efficacies. So here's the difference. So what's the difference between the different PD-L1 VEGF and the molecular structure? You see the design is different. Because of this molecular difference, and you can see their efficacy is different. So the ED50 is very low, very high potent. And we tested the compound in the cell models. And in some of the cell models, for example, in like small cell lung cancer, we actually showed the better efficacy than AK112. And we also showed the efficacy combo with chemo and the widest like ADCs. So we tried 3 different ADCs, which is already approved or in the late stage. And we show they all have like better than the ADC or by the monotherapy itself. So which enable us to move into the clinical trial. So in the monotherapy, we did finish the phase 1A, 1B. Now is in the 1C, which is like efficacy expansion stage, also like phase 2 studies. So total we have 36 patients. So we didn't reach the MTD yet. And at the R2 PD dosage, 20 milligrams per kilo, we haven't seen the DLT. And for the TRAEs, we only had 13.8, which is much lower than the AK112 and the PM8002 at the phase 1. And we also see the promising efficacy in phase 1 studies, because we see, like for example, in the cervical cancer and the lung cancer and kidney cancers, because in this stage, we only recruit 1 or 2 like in our patients in each cancer type. We see the like PRs and also the SDs. So in the combo, so we get the approval from the Chinese authority go through the combo, and we probably had the widest combo design. So we designed for combo with chemo and also the 3 different ADCs. So far, we recruited like patients in the chemo arm and also the like in our 264 SKB 264, and with a few patients, we already see the like in our safety. There's no actual safety issues, and also the signal for the efficacy is we already see in 8 weeks treatment. We already see the like in the PRs. So we see the takeaways. So our compound is clinically differentiated. We have most extensive combo with ADCs, and we see the rapid onset. So what we are here to seek a partnership for the potential like investment and BDs. We welcome like co-development or the like new co-opportunities. Thank you very much.
Thank you. All right, for this last one, can everyone please remain seated? This is our very last one, so let's give our full attention. Thank you. Last one is Kun Meng from Zhejiang Pharma Group.
Apparently, I'm not Kun Meng. I'm speaking on behalf of him because he cannot be here today. How to get to it? Okay, I'm Cheryl Lee from Pharmacokinetic. I'm a strategic partner to a lot of Chinese companies to help them to globalize their products and also helping the overseas companies to take their products to China. So today I'm going to introduce one of my partners, Dr. Kun Meng's company called Synergy. And Synergy is not new to a lot of Chinese audience here. Let me say a few words about the company first, and then I will focus on introducing several of their products around carotene, and then talk about partnership opportunities. The company was founded in 2006, which is around 19 years old, and it went through a long way to come to commercialize their first product. Now they're at a stage, their products are on a schedule to be launched. I think it's a good time to bring them to overseas for us to look at if their products can be commercialized outside of China to benefit the patients globally. They're focused on cancer and autoimmune diseases, and they have several products are approaching late-stage clinical trials. I will talk about them later. And very importantly, they focused on making sure their IP protections on several of their plant-based products are well positioned for global launching. And they have a very experienced team. I will show their faces to you. And very importantly, they have very strong financial support over the years. And that's why they can do what they're doing today. And this is the two faces I picked to show you. They were acquainted at Harvard Medical School, and they started to work together about 20 years ago. And many of the management team, they were either co-workers or classmates, worked in the Western countries for years. And very importantly, their scientific board and a lot of their teams, even they came out from the companies over the years, because innovation is a long journey. A lot of them find other ways to run their business, and they came out, but they still are stakeholders to company. They helped a company to grow together. And this is a show of their end-to-end value chain, all the way from the plant seeds breeding to the manufacturing of the finished product, and the commercial teams. They have well positioned to really take a few more products to the marketplace. And this is a snapshot of their product pipeline. And the first few, you can tell, based on 162, there are carotene-based products. Either they are used alone or in combination with other products for additional therapeutic indications, or they are the new formulation based of existing products to give them better efficacy or safety profiles. And they are into other modalities. I will talk about them briefly later. So this is a company with a lot of innovative products, but they're very low-key. They only focus on science in the past 10 years. Because they are financially pretty strong. And you can see over the 15 years in their span of the company life, they already raised 180 million US dollars. And many of the big name investment firms are behind them to support them. And they're on way to submit IPO in China. And they had won a lot of awards because they are uniquely positioned to use traditional Chinese medicine, the plant, the herbs, as starting points to make the innovations. So the first product I will speak to you about is a product that is already launched in China. It's a carotene soft capsule for HCC. And a few speakers earlier already talked about the importance of having other therapies for HCC, because either they're too expensive or they are not efficacious or they have safety profile issues. So there is a need to have better medicines for HCC, especially for late stage HCC. And this carotene is isolated from the Chinese traditional medicine, epiderm, and it's also called horny goat seed. And a lot of people are familiar with it. It's used traditionally for male vitality and arthritis, marrow and bone problems. And this is proven in TCM arena as being very effective and safe. And now Zhejiang had isolated a pure individual compound, and is now a single component compound that can be filed in other countries that are not familiar with the combination components like TCM. This part I will skip, and the other part I will speak about the advantage of carotene. It's really a very convenient dosing formulation. It's a soft gel, oral, active, and safety profile is great. And also it can work with other therapeutics together to make the treatment much better than before. And here is the development history. All the way from 2006, they started to work on this project. As you know, at that time, China is not really well positioned for doing truly innovative product, and they are the first one to do that. And as a result, a lot of awards were given to them for their exploratory work. And this product is proven by many scientists and themselves. In Zhejiang, it went through a lot of different mechanisms to modulate cancer through different pathways in here. And also uniquely, it also regulates the tumor microenvironment and make it very unique to suppress proliferation for cancer cells and also improve the immune response by the patient itself. And this is a snapshot of clinical study because it's approved product, so I will not go through how effective it is. It's very effective and safe, so it's approved. And also it's on the China's, I think over here, the National Basic Medical Insurance drugs because it's cheap and accessible. And now I will speak about the combination application. I will be very quick. I will be very quick because they do have a lot to talk about. I want to make sure it's well introduced to the audience worth your time. So this part is the most important part. The combination of carotene with the gold standard treatment for the PADC, the pancreatic cancer is very interesting, and the results showed right here. I won't talk about the mechanism. Results shows here. The ORR is 60%, DCR is 95%, and all the experts in the audience know what that means. And the median DOR is about 7 months. This is a 20-subject ITT study done at Zhejiang University Medical Center. The key PIs are very excited about this result. About 35% of the late-stage patients went through one cycle of the treatment. They were able to do surgeries, and prior to that, they were not able to do surgeries. So this is a great improvement and exciting results. Other things I will not talk about, they are planning to do clinical studies, the formal clinical studies with this application and also CRC application. And they also have a synthetic molecule based on acarotene. This is also ready for clinical studies, and in vitro study results are very promising as well. So in a snapshot, this is a company with a very experienced innovation team, and they have really persistence in doing innovation. Now they are mature to a time, I think they can really collaborate globally to bring those products to other markets. And the product itself is already approved for one individual application. Now it can be used in other therapeutic areas. And also, I think very importantly, this is a very inexpensive product, easy to manufacture, easy for patients to take, and it has a lot of good things about it, and I'm happy to answer any other. Battery is gone. I'm still here. Thank you very much.
Thank you very much. All right, thank you to all of our speakers, and thank you to everyone here staying with us in the audience. We have food on the first floor, so please help yourselves, and also on the third floor if you're a paying attendee and also speaker as well. You can come join us at the VIP room. And there's also line dancing happening at 8:30, so make sure you don't miss that. Thank you very much, everyone. Thank you.